The overall goal of this procedure is to define microRNAs that estrogen receptors may regulate. This is accomplished by first preparing the cells for maximal estrogen response through culture to an optimal confluence and depletion of estrogenic activity. The second step is to induce transcriptional activity of the estrogen receptors by treatment with estradiol for the required number of time points. Appropriate controls are included for comparisons. Next, the RNA, including the microRNA population, is extracted and converted to cDNA. Known estrogen-regulated target genes are then measured using qPCR to confirm that the treatment protocol was successful. The final step is to determine the genome-wide microRNA expression pattern using large-scale screening technologies. Ultimately, confirmatory analysis using qPCR is performed.
This method can help answer key questions in the breast cancer field, such as whether and how estrogen affects microRNA expression.
Though this method can provide insight into breast cancer, it can also be applied to other systems, such as other cancers or cell types that express either estrogen receptor alpha or beta.
The MCF7 cells used in this study are prepared 5 days prior to treatment. In a sterile hood, remove media from the T75 flask using a sterile pasture pipette connected to a vacuum. Gently wash the attached cells twice with PBS and detach the cells by trypsinization. Count the cells using a cell counter according to the manufacturer's protocol.